Hello everyone. In this video we will discuss principle and some basics of chromatography. The chromatography is based on the principle where component present in the mixture passes through the stationary phase with the help of mobile phase. This stationary phase separates molecules depending on some factors. These factors are adsorption, partition, affinity, and differences among molecular weight, and of course polarity. There are two phases involved in this separation: stationary phase and mobile phase. Stationary phase, the component of stationary phase are usually solid and sometimes liquid. The component of mobile phase is always liquid or in a gaseous state. For example, if we look at if we take the example of column chromatography, this is the column. Column is made up of glass or stainless steel in which stationary phase is incorporated. There are different types of stationary phase depending which what we want to separate whether it is our mobile phase is polar or non-polar. So stationary phase is incorporated into the column and mobile phase passes through the through this column, and separation takes place and the collected sample is called eluate. There is another factor which is called distribution coefficient and partition coefficient. Distribution coefficient or partition coefficient refers to the concentration ratio of a species present in the compound. For example, if we want to calculate the distribution or partition coefficient is equal to the, if we have two phases, phase A and phase B, so concentration in phase A upon concentration in phase B is equal to the distribution and partition coefficient. Basically it is a ratio of concentration between two phases. Another is retention time. Retention time is it's a amount of time a component spent on the column after it has been injected. Means the time spent by a component in the column, where suppose this compound passes through the column, the time spent by the component in the column is recorded as retention time. This retention time denoted as small t, capital R, TR. So another factor is retention factor. Retention factor or RF. Retention factor is used to compare and identify compounds. It is the additional time or distance traveled by component. Means that time spent by column is denoted as retention time, but the extra time and extra distance traveled by the component is denoted as retention factor. It is used to compare and identify compound. RF value is equal to the distance traveled by solute A upon the distance traveled by solvent. If in case of thin layer chromatography, suppose this is the spot of a mixture and it after some time when the stationary phase passes, this is the example of thin layer chromatography. In thin layer chromatography, when the this spot traveled a particular distance, this is denoted as A, and the distance of the solvent is denoted as B. So when we discuss thin layer chromatography, we will discuss in detail. So these are some important points regarding the chromatography. I will repeat again. The chromatography is based on the principle where component present in the mixture passes through the stationary phase with the help of mobile phase. This stationary phase separates molecule according to some factor. These factors are affinity, partition, and absorption, and differences among molecular weight, polarity. So depending upon these factors, the separation takes place. There are two phases involved in this separation. One is stationary phase and second is mobile phase. Stationary phase made up of usually solid or a liquid, and the mobile phase is always liquid or in a gaseous state. There are some other important factors: retention factor, retention time, distribution coefficient, which helps us to understand the principle of chromatography. So what is distribution coefficient? Distribution coefficient is the concentration ratio of the component between two phases. And what is retention time? Retention time is the amount of time spent by a component in the column, and when additional time what is retention factor? Retention factor is used to compare and identify compound, but this is the additional distance and time is traveled by the component present in the mixture. RF value, we calculate RF value in case of paper chromatography, thin layer chromatography. So RF value is equal to distance traveled by solute upon distance traveled by solvent. Thank you very much. I hope you understand. In the next video, we will discuss types of chromatography.